for the invitation. It looks to be a great series, a great workshop this week, particularly. So I find this a very frustrating time in population genetics, evolutionary genetics, because we have a huge amount of data. In principle, we have the possibility of getting complete sequences of every individual in a population. And there's clearly a lot of information about both demography, demographic history, and about selection locked up in that data. And we have a very simple, elegant kind of theoretical framework, at least for the neutral case, where we have the ancestral recombination graph describing the coalescence and recombination of lineages, genomes, as we go backwards in time. And in principle, we should just be able to put together this very fine theoretical framework with the mass of data and answer all kinds of questions. But it's kind of a little bit embarrassing, actually, that I don't think we've really answered the basic questions that we started out with even 50 years ago. What maintains variation, how many loci are selected, what's the distribution of effects on quantitative traits, and so on and so forth. And we're using really very, very crude methods for data analysis, which are clearly throwing away a lot of the information that we have. So at the simplest level, we have the idea of looking at individual SNPs one by one. So if we had a sample of genomes from a single population, we could just count the site frequency spectrum, the numbers of copies of a particular variant at a particular site, and that will tell us a lot, and we can treat the site frequency spectrum across the whole genome. But that throws away any information about associations between markers, between SNPs. We could divide the genome up in a rather arbitrary way into blocks, which we imagine are not recombining too much, and we could try to estimate the genealogy connecting, well, I suppose I could write like that, the genealogy connecting the particular set of blocks there. But again, that's a rather arbitrary procedure, and it's again throwing away any information about recombination, about linkage disequilibrium. And of course, there are all sorts of methods out there, but all of them are partial ways of looking at the data and somewhat arbitrary. And we'd like, in principle, to be able to calculate the probability of observing the set of mutations that we see along a linear genome, given the ancestral recombination graph, which is, at the moment, a very distant dream. So what I'm going to do is talk about really a very simple, straightforward, very general method for calculating the probability of a genealogy at one or a number of loci, possibly in a structured population. And this is work that I've been doing with Konrad Lerzer from Edinburgh, who I hope is here and is there to answer, yes, difficult questions. Because I'll also rather briefly mention applications that Konrad's been making to various data sets of this approach. The idea of calculating exact likelihoods for admittedly rather small data sets. We can manage, 3, 4, 5 genomes maybe, but genome-wide. And then what I'll concentrate on at the end of the talk is a generalization of this machinery, really, for calculating the joint probability of genealogies and recombination events along a continuous linear genome. And I have to admit at this stage that I haven't quite been able to get to the last sentence of the abstract, so don't read the abstract in the book, but there are going to be interesting applications, so I'll outline this as exercises for the reader at the end. I'm sure we will want something to do in the other talks. The basic idea, though, is that we should be able to use information from the patterns of change in genealogy along the genome, using essentially recombination as a clock to give us more information about timescales, about rates of processes, and actually to give us a clock which applies over a much wider range of timescales than mutation can. Okay, so mutations occur at a set rate per site. Recombination, of course, occurs at for reasons, I guess, which are rather interesting but unclear. Recombination occurs at a similar rate per site, but of course, if we look over larger and larger ranges, we can estimate co-ancestry over a whole range of timescales. And for example, there's very nice work by Graham Coop, who I think has been substituted here. Graham's not here, as you know, but he sent a substitute. Okay, good. So it's nice work looking at at identifying individuals who share ancestry at some time in the past on the grounds that they share lengths of genome which are identical by descent, and the longer the stretch identical by descent, the more recent the co-ancestry. And so you're using their recombination rate as a clock to tell you the time since co-ancestry. And the idea is generally that we should be able to use much more effectively information about patterns along the genome, along a linear continuous genome, using as a fundamental model the ancestral recombination graph. Okay, so before I get on to that, start with the basic idea behind all this, which is that we can describe a genealogy by the lengths of the branches. And so if we have a set of 3 genes, A, B, C, I'll denote the complete set by omega. And so now we have here, in this particular topology, we have four internal branches described by TAB, TA, TB, TC. That is the times of the branches leading down to a certain set of genes, either one gene or, in this case, these 2 genes. And in general, we can describe all the possible topologies by listing a vector, in this case of 6 possible branch lengths, which encode both the shape and the actual lengths of this genealogy. So then we say that instead of working with the probability of observing a certain set of branch lengths, we work with the Laplace transform of that, or the generating function for that, which is just given by this expectation. Okay? And from that, we can calculate anything we want. And in particular, we can calculate rather easily the probability of observing a certain set of mutations. So we've transformed the probability of a certain set of branch lengths to a function, and it's a function of a set of dummy variables, these omegas, which correspond to the branch length. So for every branch, we have a corresponding dummy variable omega, and we generate this function, and we want to calculate things from that. For example, we might want to calculate the probability of seeing a certain number of mutations on a particular branch. Call that Ks, the number on the branch leading down to the set S of genes. And basically, you can calculate that just by differentiating the generating function a certain number of times, by a number of times equal to the number of mutations. And so we can calculate observable quantities, mutational configurations. You can calculate in the same way the probability of seeing a certain set of mutations, a certain number which are n. So, for example, this particular mutation is shared by A and B, so we will denote that KAB. This one is shared by A, B, and C, and so on. We can calculate the probability of seeing one of these, and so on, by differentiating this generating function. And that's relatively easy to do, as long as you don't have too many mutations. Okay?
Is there a question?
Yeah. Is there a question?
Yeah. I don't quite get your notation for, how are you coding different topologies here?
Okay, so that's the neat thing, that this single function codes all of the topologies. So this particular case has constraints on the branch lengths, right? Which mean that TAB is some number, TAC and TBC are 0, okay? And so on, and then TA equals TB, etc. But there's also a probability of another topology which will have other constraints. And all of that translates basically into this generating function, which actually is a sum of terms, and each term could be identified with a certain topology. Okay? Okay, so we want to be able to calculate this, and it turns out that there's a very simple recursion for the generating function, which can be automated in a rather straightforward way. And what we do is simply trace back from a certain configuration of genes. Here we've got 3 separate genes. We trace back to the next previous event, which is, in this case is a coalescence between A and B. And in this very simple example, a single population with 3 genes, there are 3 possible events we can have. A coalescence between each of these 3 pairs. And we could generalize it. We could have multiple loci, for example. We might think of having the possibility of recombination as well. So let's say we had 2 loci. We could have, let's say, samples where we took initially 3 chromosomes. At this locus we have A, B, and C labeling these alleles, X, Y, and Z labeling these alleles. And so that we have genomes which are ancestral to A and X, and the 2 loci have B and Y, C, and Z. And as we trace back, well, maybe they're inherited together and they coalesce together. But maybe there's a recombination event such that this lineage then traces back to there. So now as we go back, we can count a rate of 2 kinds of events, recombination events or coalescence events. And the recursion turns out to be very simple. It's just basically a sum over the possible events, a sum over the generating functions for simpler configurations in which these have coalesced, for example, plus a sum over recombination rates times the configurations in which these lineages here, or this lineage here, split into 2 lineages, one ancestral to C and the other ancestral to Z. Okay? So we're, in this term, simply summing over all possible events as we go back in time. And then we divide by this term, which is related to the total rate of coalescence, which is just k choose 2, plus the total rate of recombination, plus a sum of dummy variables corresponding to these leaves. So these dummy variables in this example would be WA plus WX plus WB plus WY plus WC plus WZ, so on. That is, we sum over all the dummy variables corresponding to these lengths of these lineages. And the reason for doing that is that what we're doing as we go back is simply adding an exponentially distributed time to each of these lineages. And this generating function has the property that if we add an independent exponential variable, we simply multiply the generating function by the corresponding function, the Laplace transform for that exponential distribution. So we have this very simple recursion, which, as you can see, can be rather easily automated.
So this is for constant population size?
Yes, well, yes, it is, right. But no, I did say it was general, so it's quite general, I suppose it's. We'll get to that, yeah. Yeah, so I should say, so far, it's really been very, very simple. Standard coalescent, scaling time relative to 2n, I should have said that. So this is the standard coalescent, but you can actually have a very, very broad view of what events are. Here I included recombination. But we could also say that there are changes in population structure. There is a certain rate going forwards in time at which populations split. Okay? And there's a kind of neat trick which says that if we calculate the clear some space. If we calculate the expectation of this quantity, we end up with something like term involving e to the minus lambda t, where lambda is the rate of population splitting. And if we then take the inverse of this generating function, which includes this rate of splitting, treat this as a Laplace transform, we take the inverse Laplace transform, we can actually calculate explicitly the generating function given a certain event at a certain time in the past. So there's a way to go from a model in which we include a uniform rate of various events, recombination, coalescence, and possibly population splitting, or possibly the existence of a bottleneck or something like this. And then we can take a rather simple inverse transform with respect to the time of the bottleneck, the time of the population split, and calculate the actual probability for a structured population. Sorry? Yeah.
I have a question. Is this dummy variables that you mentioned, the indeterminates, are they corresponding to the lengths or to the specific tree structure?
Okay, so...
Because if
What's the question?
Yeah, yeah. So the question was, what are these dummy variables? So in general, if I just go back stage, I probably can't actually read it back, but these are vectors. So this is the vector of dummy variables dot product with the vector of branch length. So there's a dummy variable for each branch length. So you have complete information about the whole branch structure. If you're only caring about, say, the length of the genealogy, then you just set all of these to the same value, all the omegas to the same value, and then you get a single function of a single variable omega, which gives you the generating function for the length of the genealogy.
But there are many ways of writing this for different lengths. I mean, I can split the length in different ways, right? I don't think it's well defined in this way.
No, I mean, you have a genealogy like this. So if you tell me, well, if you know what these branch lengths are, then that gives you all the observable information about this genealogy.
Condition on this is well defined, yeah.
Yeah.
But in general, for this specific tree structure, maybe I'm missing something.
Yeah. Well, and then you also have the possibilities of other tree structures, but those simply have a different relationship amongst these branch lengths. So here, you know that it's this topology because TBC and TAC are 0. Okay, so that, you imagine a distribution over all possible genealogies, and there'll be a distribution for this topology, this topology, this topology. And each of those depends on a smaller number of branch lengths. But in aggregate, there's a complete joint distribution of branch lengths, which covers all the topologies. Okay. Anyway.
For what the dummy variables are, I mean, intuitively, I like to think of them as rates.
Yeah.
I mean, I know that's not exactly.
Yes. I don't know. I've always thought of them as just dummy variables. So you know, there is a procedure. I don't know. Maybe you have better intuition for Laplace transforms because you did physics properly, right? So you can see the Laplace transform in your head. But yeah. Okay. So I'm not actually going to say very much about this population structure issue, about structured populations where we have migration, we have splitting of populations, we have bottlenecks and this kind of thing. But just to say that the method does apply to that. I'll be focusing later on the issue of recombination and extending this from discrete loci with discrete recombination rates to a linear constant. But before doing that, and before running out of time, I'll just briefly mention the work that Conrad's been doing to apply this scheme to some data sets. One on gall wasps, which are kind of nice to work with because they're haplodiploid, so if you look at male, you've just got a haploid genome, which is convenient. And this particular paper was based on 5 gall wasps, 5 male gall wasps, 1 from Iran and 2 from Central Europe, 2 from Western Europe. The data was reduced down to 2 replicate data sets of triplets, east, central, west, based on 2 almost different sets of 3 gall wasps. And the question was, can we distinguish different models of population history in which we have different schemes for population separation, different schemes for admixture between those populations? And to cut a long story short, you can, using this scheme, estimate the best supported model and estimate the degree of admixture between them. So the best supported model in this case is that the eastern population split from a central population and then further back in time had split from the western population. And there was evidence of a recent admixture event from east out to west. Okay? And you can estimate actually rather precisely the times of these various events and the proportion of admixture. The dotted lines and the solid lines here correspond to the 2 replicate data sets, which correspond just to different flies chosen from more or less the same populations. And this was based on about 2,000 blocks of genome, arbitrarily defined to be 2 kb at least long and to be well separated. So you have complete genome data, then filter that, clean it up, and get well-aligned blocks which are reasonably well separated. But actually the results are rather robust to the numbers of, to the sizes of blocks that you choose. So what I should say, that the scheme is based on a method which is very nicely scalable to large data sets. So the idea is that for a small sequence block with not too many mutations in it, you can tabulate all the possible mutational configurations and calculate their probability. Okay? And obviously you can't do that for an indefinitely large number of mutations because that involves differentiating this funny function many, many times, and the computer collapses. But you can calculate the probability that there are 0, 1, 2, 3, many of each kind of mutation and tabulate a big table of all the possible combinations of those up to some upper limit. And then you simply multiply the likelihoods across all the loci. So there's really no limit to the number of loci you can have. The limit is in the number of configurations you have to do the calculations for. Okay. So just to mention a second analysis, which Conrad has done with Laurent France, who unfortunately was supposed to be here but can't be here. He had to leave early. But this is on the Neanderthal data set. In fact, what they're aiming to do is to do a similar analysis for a much more interesting situation with pigs in Indonesia, I think. But they're warming up with the Neanderthal data set. And they basically use the same method, using this idea of dividing the genome into short blocks with a small number of variants, and really trying to analyze the data more efficiently than simply with the Abba-Baba D statistic, which simply compares the numbers of two different kinds of SNPs corresponding to two different discordant genealogies. They go beyond that by asking about the complete distribution of all the different mutations that you can get in short blocks. And by doing that, they definitely reject the hypothesis of ancestral structure and estimate a rate of admixture between the Neanderthal and the European population of a few percent. Okay? So it's giving the same kind of picture as the D statistic does, but it's doing it much more accurately. And the accuracy seems actually rather surprisingly to us to be quite robust to your choice of block size, to the presence of recombination, which actually has been ignored here. Yeah.
So the mixture proportions are discrepant with the current estimates of mixture proportions. So, which is maybe fine. What regions of the genome are you looking at?
Conrad. There you are.
That's the whole also.
So you're using the whole genome?
The whole lot, yeah.
So there's a discrepancy. That's fine.
Okay, so yeah. So what's the current estimate?
I think we'd be very surprised, based on our last paper, if it was more than, I mean, most of the estimates were under 2% with standard errors that would exclude numbers more than 2.5%.
Right. And is that based on D or on?
It's based on something like a D statistic,
yeah,
basically the numerators of the D statistic, an F4 statistic ratio.
Yeah. Yeah, we can probably talk later about that because it's, I mean, or rather Conrad can talk later about it, but it's, I mean, one of the issues, of course, is that you're getting extra information here from singletons, and there's a lot of error in the Neanderthal singleton because of all the error. So there's some issue about how you clean up those data.
You're using rich information that was not used there, so it's very interesting.
Well, anyway, yeah. I mean, maybe if there is a discrepancy, it points to something real, that some other model is appropriate and not the models we're looking at. So. Okay, so...
How does the... Sorry, another question. How does the approach relate to other methods that have been used to do similar things, like Zihang Yang's or method, or MCMC call, or
okay, so the MCMC methods, as I understand it, are really very hard to scale up to very, very large numbers of loci because you have an extra computational cost for every locus you add. And so actually it's very impressive that they work with a number of loci they do, but it clearly scales much more slowly than this does. Okay? It's quite close to things that Zihang Yang's done, and in fact there have been calculations around. Also Oscar Hobolt has done calculations of exact likelihoods and used these kinds of methods. And I think what I'm most interested in is that it's a very general method that allows you to do the calculations in an automatic way for complicated models. You know, if you try and do it by hand, you kind of go mad after a very short time. But here, at least, the computer is driven mad after a slightly longer time. Right? Anyway. Okay, so I'll just, as I've not got too much time left, when do I end? Is that, I've got 10 minutes or 15 minutes? Oh, well, I can do the whole genome. Right? Okay, so this is all still very crude, because these analyses are sort of taking a continuous sequence and arbitrarily chopping it up into bits that you hope aren't recombining too much, which actually can't exactly be true. So what we'd like to do would be to have a way of generating probabilities for a truly continuous genome. And the situation we have then is that we have this long line, we choose a random point on the genome, and let's work out in one direction. Okay? It's easy to see how you generalize it to two directions. Work out in one direction. At some point, there will be a recombination event that changes the genealogy. So let's suppose that we actually have 3 sampled genomes, and at the focal point, there is a particular genealogy, let's say like that. And so this is purple region. And then we move out, and at some point there's a recombination event somewhere in this genealogy. Let's say there, and a coalescence back there. So that the topology is the same in this example, but the coalescence time between these two is slightly shorter. Okay? And so we now have a region with a slightly different genealogy here. And then we carry on, and we have now maybe a recombination event here. So we might have another recombination event there, so that now this red genealogy, if I've drawn this right, looks like this. Okay? So what we really want is a joint distribution of genealogies which are being described by these vectors of branch lengths and locations of recombination events. Actually, what we work with are the sizes of these intervals. And so we want to find some kind of generating function for the joint distribution of genealogies along the genome and positions at which recombination events change the genealogy. Okay? And there's a list of subtleties which I've put here, which are probably, well, just to indicate really that there's some tricky parts to this. It comes up with a remarkably simple formula, but there's some tricky issues to deal with. The basic reason we can work with it is that the distribution of these distances here are closely related to the lengths of the corresponding genealogy. Because basically, a recombination event can occur, if we talk about this first recombination event, that recombination event can occur anywhere on the focal genealogy, on the first genealogy. And the probability that it occurs as we move along has rate L0, where L0 is the length, total branch length of the first genealogy. And then as we move on, we now have this green genealogy. We can now get a recombination event which occurred here. And again, that's distributed over the genealogy. But now we actually have potentially we have actually more branches on which it can occur. It's slightly tricky. I won't go into all the details, but basically as we extend the ancestral recombination graph, the recombination events can occur anywhere on the genealogies proximal to this. So that if we go out to, say, Ln here, a recombination event could occur and could change the genealogy anywhere proximal to that. But the total length of the branches is not just the sum of all those lengths, because most of the branches coincide. So we define something called L star, which is the length of branches on which a significant recombination can occur, basically. And the basic point is that recombination in non-ancestral material can be important. So that if we had, for example, a situation where a certain lineage was carrying a block here ancestral to A, and another block also ancestral to that same genome, then a recombination event here, even though it's not in ancestral material, will produce 2 lineages, and those will then behave differently if we start thinking about coalescence with some other lineage. Basically, there's twice the rate of coalescence of a lineage with 2 recombined lineages than with a single lineage. So that the precise probabilities do depend on kind of internal structure, depend on events that occur actually anywhere in a longer and longer stretch of genome. Another complication is that we can have, rather annoyingly, recombination events that coalesce back, that produce a lineage that coalesces back onto the same lineage. And then we have two blocks, as I've defined it, but they have identical genealogies. And we just have to live with that. At least I haven't found a way to produce a simple recursion which guarantees that these points are points where we have distinct genealogies. There's a small probability that actually these happen to be identical genealogies. But that's not too big an issue. A final point to mention is that it matters that we choose a focal point at a particular location. We throw down a random point here, and we have a certain length of block out to here. But what we find, and it's sort of a little bit puzzling at first, is that the distribution of lengths here is different from this distribution, is different from this distribution. There's an ascertainment issue. The point is that if we take a series of blocks defining different genealogies along the genome, and then we throw down a random point, that random point will tend to land on a long block, which will also tend to have a shallow coalescence time, a recent coalescence time. And so the distribution of coalescence times at a randomly chosen point tends to be, get it right, tends to be shallower than the distribution of coalescence times at a randomly chosen block. And therefore, as we work out from a random location, we find actually that the blocks get shorter. Okay? So, I'll just skip quickly through the case of two blocks. And the basic idea is very simple, that the probability, the joint probability of the branch lengths, T0 and T1, and the locations of the recombination events, chi0 and chi1, just depend on, in this simple case, the lengths of the genealogies. And we follow a generating function defined in this way, not quite the generating function of this joint distribution, but very close to it. And we can derive a recursion, which looks actually very similar to the one before for a discrete loci, in which we have a denominator. Let's put the parentheses in. Yeah. We have a denominator, now it's correct, in which we have the total rate of coalescence, the rates of recombination, k being the number of lineages that can recombine in this simple example. But these are now multiplied not by recombination rates, as before, but by the lengths of the intervals, chi0 and chi1. These are the intervals, sizes of the blocks, basically. And then on top, we have a sum over possible coalescence events, possible recombination events. But we have another trick that we have to keep track of. We have to have an index here which says there is one recombination event pending. Because if we've decided we're going to follow these two blocks separated by a recombination event here, then once that recombination event has occurred, it can't occur again. We're restricting ourselves at the start of the thing to looking at just two blocks separated by one recombination event. And so once we have made the first recombination event, we now say we use psi0. Sorry, this is the recombination event happened. We now go to psi0, which is the standard coalescent with no recombination. Okay. So, So in this example, we can write down the generating functions. The generating function here for two blocks and two individuals. That can only change by recombination, because if we allow them to coalesce, we then wouldn't be allowed to have a recombination. Everything would have ended. And yet we're saying we want to have two genealogies separated at locus A and locus B, separated by a recombination event. So we only allow recombination, and it can occur either between B0 and B1 or between A0 and A1. Once it's occurred, we then go back to the standard coalescence psi0, which is given by the expressions for a standard exponential distribution and so on. So it's a very simple expression here. But the result we get is actually non-trivial. We can calculate the, for example, things like the distribution of lengths of blocks sharing the same genealogy. And interestingly, the distribution of lengths of the first block, that is the position from a focal point to the first recombination event on the right, is a very fat-tailed Cauchy-type distribution, which has a mean of infinity. So that's actually a bit depressing from the point of view of data analysis, because it tells you that if you land on a random point, you have a small probability of having an extremely long region which will share the same genealogy, implying there's going to be a lot of sampling error, a lot of error, depending on where exactly you land. It's a distribution of an infinite mean. However, the distribution of lengths of the next block is better behaved. It's kind of complicated form, but it has a mean of 0.88 scaled relative to population size. And that's simply because of this ascertainment issue. The first block you land on is likely to be longer, maybe much longer, than a typical block. And let's get over that. To cut a long story short, we can extend this to actually get a generating function for an arbitrary number of blocks, for an arbitrary number of genomes. And we have to actually track an index here which keeps count of the number of recombination events we've used up. So we might have 10 blocks, 10 recombination events, say. So then we have a vector which ticks off as we go through the recursion that we've had this recombination event, we've had this recombination event. And it's a nice recursion because it's closed. We simply iterate and we get down to the answer. Okay, there's nothing complicated about it. And that means that we can automate it. So I hope this works. We can define a function which is only about that much code. It's pretty simple, which actually produces a set of equations telling us what happens after one round of the recursion, after one event. Given a generating function for some configuration S, whatever that is, might be a configuration like that. Then we iterate it and we express it in terms of a linear combination of other configurations. And we keep going using this fixed point function until we end the recursion. And so it's quick, right? So it gets, of course, a lot slower if you add more and more genes, but you get a function here which encodes the topology, which encodes all of the branch lengths of the two genealogies. And go down here. It's fairly short, isn't it? Go down. Had a bigger screen. We can simplify it by this operation simply collapses all the dummy variables. So we say we only care about the lengths of the first one, the length of the second one. So this is actually the generating function for the joint distribution L0, L1, the two lengths of these, which corresponds to the numbers of mutations you would see in the first and second genealogy. Back. And I suppose we can check that this integrates to 1. This is unwise. No, it doesn't. Oh, well, mathematics has broken down. That's a shame. Well, let's ignore that. It does integrate to 1 on good days. OK, so I've sort of run out of time, but basically you can do lots and lots of calculations of things like the probability that there are no SNPs in a certain interval, length x, and you do that by saying, what's the chance that the block is longer than the interval x and that there's no mutation? Then what's the chance that there is one recombination event within the interval and there's no mutation in either of them, etc. And for a relatively low rate of, so up here for mutation to recombination 0.5, that is recombination twice as fast as mutation, as you go in from a calculation based on a single block, where you can put bounds on the probability of seeing no mutation per distance x, to 1 block, to 2 blocks, to 3 blocks, and so on, you converge on a good estimate of the distribution of intervals with 0 mutations. If you do it for a lower recombination rate here, high mutation rate, you have more information. You converge extremely quickly to the distribution. So at least if mutation is fairly fast relative to recombination, you can do calculations with this scheme by looking at successively more and more blocks without running into computational nightmares. So this is a similar sort of exercise in which we look at the probability that if we have 3 genomes and we have up to 2 recombination events, what's the chance that we get a change of topology? So we start here with, let's say, one genome has A, P, X, another genome has B at this locus, Q, Y, another genome has C. Okay, so this is one locus, another locus, another locus. And clearly, we can have various topologies, 3 different topologies in principle. We can ask what's the chance that these 3 blocks have the same topology, or that the topology changes at the second site, or at the first site, or it changes twice, and so on. And you can also then calculate the expected distance, expected sizes of those blocks conditional on the topologies. And these are the sorts of things you might imagine trying to use in data if you were starting to observe discordant or concordant topologies as you move along the genome. But, go on. The question, of course, which probably has occurred to those people who are not already asleep, is what you might actually do with this machinery in relation to real data. And the closest I've got to it in this talk, I hope to get a bit further, but I didn't, of course. The closest I got to it was this issue of the distribution of lengths of interval in which there are no mutations, or the distribution of intervals between SNPs, essentially. And that's something you can calculate with this machinery. But the sorts of things I'm thinking about doing with it is, first of all, to ask, how robust is this method of assuming a non-recombining block? Suppose we have bit recombination. How far does that bias or alter your estimates? Another issue is, how accurate is the very popular and convenient assumption of the coalescent as a Markov process along the genome? I mean, it's clearly much easier to analyse data if we can assume that the genealogy here only depends on the immediately adjacent genealogy, and we treat the process moving along the genome as a Markov process. That makes things far simpler. And if you do calculations for discrete loci, 1, 2, 3, 4 loci, then you find that it fits pretty accurately. You can calculate joint distributions of coalescence times and so on for a number of loci, and it's reasonably close. The difficulty, though, is having faith that somehow these errors won't accumulate when we're analysing tens of thousands of loci. Now, it's not immediately obvious to me that one can trust this Markov assumption. Maybe it's fine, maybe it's not, but it would be nice to use this kind of more analytic approach to test the accuracy and robustness of the more approximate methods, which we're going to have to use for larger data sets. Okay, thank you.
Maybe following up on the definition at the beginning and the question at the beginning about how to incorporate, for example, demography or like
yeah.
So, I mean, if we thought about the non-recombining region, all the recombination terms would cancel out. But how would you integrate, like even with that, like into this generating function? How would you incorporate like a bottleneck or something?
Okay, yeah. So, well, it's probably easier to explain the population splitting argument, but it's basically the same for a bottleneck. So you can easily put into this model anything that has a uniform rate. So let's suppose we say, instead of that there was a split in the population at some time, definite time in the past, we say, well, there's a certain rate at which the population splits. In other words, we don't know when the split occurred, and we have some prior, if you like, with an exponential distribution of the time when the split occurred. So actually, it's not clear to me that that isn't any more reasonable or unreasonable than assuming a fixed time. It's another model. So we can then use the recursion, and we have a formula for the generating function which involves this rate of splitting. But if you think about it, that generating function is actually the Laplace transform of the time at which the split occurred. And if you just take an inverse Laplace transform with respect to just that one variable, which actually is not that hard to do, typically, then you can find the actual. I mean, you tell Mathematica to do it. You just put inverse Laplace transform, you put the generation function in, and it does it. I don't think I could do it. Anyway. So then you get a probability of seeing the observed mutational configuration or whatever, given that there was a split in the population at some time in the past. And you can do the same thing for a bottleneck. You can say, well, we actually do the calculation assuming that there was a bottleneck at some time which is distributed exponentially. There's a rate at which bottlenecks occur. But once you have that, you can then easily convert it into a probability, or likelihood actually, for the model, given a certain definite time for the bottleneck. Okay. So I mean, it's just a little trick. The basic idea is that we can deal with more or less arbitrary, I think, structured population models. Okay.
Can you tell how the computation scales with the number of loci, the number of sites?
Yeah, badly. Yes. I mean, I sort of glossed over this a little bit and chose an example with a fairly small number of sites, loci, etc., or blocks. Basically, the generating function is a big rational function with terms which correspond to different paths through the ancestral accommodation graph. And so the size of this expression is going to depend on the total number of paths through the graph, which blows up very, very quickly. So in practice, we're limited. We can maybe, with all sorts of trickery, get up to 6 genomes for a structured population model in a single locus. Or we can get up to maybe 4, 5, 6 blocks in a single population, this kind of thing. Actually, it seems that the limiting factor is often not calculating the generating function. You can press the button and after a few minutes of labouring, the machine groans and produces this generating function. It's there, it's got all the information you want. But then if you try and calculate anything from that, like the probability of seeing this particular mutational configuration, you've got to differentiate this big function with, you know, 1 million terms 10 times, and then it really goes bad. So it's slightly frustrating. You can produce this magic function that has all the information you want about the set of genealogies, but inspecting it and tabulating the probabilities of all possible mutational configuration, that actually takes much longer.
So let's thank Nick.